We are honored to have Dr. William Douglas Figg provide our next lecture. Dr. Figg received his BS in Biology from Georgetown College, his BS in Pharmacy from Stanford University, and his doctoral degree from Auburn University. He completed his internship at the University of Alabama at Birmingham Hospital and his fellowship in drug development at the University of North Carolina, Chapel Hill. He also received an MBA degree from a combined program at Columbia University and the London Business School. Dr. Figg joined the National Cancer Institute in 1992. The following year, he became head of the Molecular Pharmacology section and the Clinical Pharmacology program. Since then, his research has focused on using pharmacologic principles to optimize the treatment of cancer and on identifying genes involved in the development of prostate cancer. He has been the PI of over 25 clinical trials for men with prostate cancer. Dr. Figg has over 650 peer-reviewed publications. He has received numerous awards and honors, including the Leon Goldberg Award from ASCPT, the Alan J. Brands Award from the U.S. Public Health Service, the Russell R. Miller Award from ACCP, the Andrew Cargie Award from ASMUS, and the Sustained Contribution to the Scientific Literature Award from ASHP. Dr. Figg is adjunct faculty at Columbia University's College of Physicians and Surgeons and serves as adjunct at several schools of pharmacy throughout the country. Please enjoy his lecture.
My name is William Douglas Figg. I run the Molecular Pharmacology section in the Clinical Pharmacology program in the National Cancer Institute. Today's lecture is the Introduction to Pharmacology, Drug Development, and Clinical Pharmacology. The definition of pharmacology is the branch of biology concerned with the study of drug actions, where a drug can be broadly defined as man-made, natural, or endogenous molecule that exerts a biological effect on some type of tissue or cell. Clinical pharmacology is the application of that. It is the science of drugs and their clinical use and focuses on the principles of using pharmacology to treat diseases. There's a tremendous need for clinical pharmacologists. If you look at the number of drugs that are prescriptions that Americans take each year, about 50% of Americans take at least one prescription drug. About a quarter of the population takes 3 or more prescription drugs. 12% take 5 or more prescription drugs. And 75% of visits to the physician involve some type of prescription. 4 billion prescriptions are filled in retail pharmacies each year. And the global market for pharmaceutics is huge. $734 billion it was in 2015, and approximately half of those sales are in the U.S. 12 pharmaceutical products sell more than $5 billion per year. So let's talk about drug development. It's expensive. Some estimate $102 million to bring one drug to market. Others estimate that is much higher, at $2 billion to get a drug to market. Clinical development accounts for 72% of that cost. It's also time-consuming. The median time from synthesis to FDA approval for an anti-cancer drug is 16.2 years, 4.4 in preclinical development, 10 years in clinical development, and 1.4 in FDA evaluation. For other therapeutic classes, this is lower, quicker. 13 years for cardiovascular, for example. So this is the way I think about drug development, all the way from the beginning to FDA. So first, we have to identify a molecular target, okay? Now, this is concentrated towards anti-cancer development, but in general applies across all therapeutics classes. So identify a molecular target. Design a compound through computational approaches or conduct a high throughput screen to find something that modulates that target. Test the lead compounds in primary and secondary in vitro assays. Synthesize lead compounds. Evaluate compounds to determine molecular pharmacology. We have to understand how it's working in the cell. For cancer, it's important then to submit it to the NCI 60 screening and get those results and use the compare analysis to understand the molecular pharmacology and what cell lines it's having activity in. Next, it's important to develop analogs, structures that are similar so that you then can compare a structure-activity relationship analysis. Lead optimization is the next thing through medicinal chemistry. Additional in vitro experiments may be needed at this point. Determine an acceptable animal formulation. Determine preliminary animal toxicology. I think it's important at this point to figure out if it's toxic in animals, assess the bioavailability, and define a maximum tolerated dose in animals. Conduct in vitro experiments in xenografts, tumor cell lines. Develop a bioanalytical method for quantitating the drug, usually LC-MS these days for small molecules or ELISAs for biologicals. Characterize the preclinical pharmacokinetics, and this is determine the half-life, the AUC, how the drug is metabolized, renally eliminated, those types of things. Determine if the compound is metabolized is very important, as then you can assess whether there could be pharmacogenetic concerns. Determine plasma protein bonding, as well as renal elimination. Develop and validate a pharmacodynamic assay, biomarker assay for predicting activity. Conduct formal toxicology studies for the FDA, GMP production at this point, and develop a human formulation. Then file a IND. Then it's phase 1 to define the maximum tolerated dose, characterize the clinical pharmacology, phase 2 to determine activity in a specific patient population, and then phase 3 to compare it to standard of care to see if there's enhanced activity. At this point, you're ready to submit to the NDA to the FDA. Cancer drugs have the lowest overall rate of success, with just 5.1% of the drugs that enter phase 1 ultimately reaching the FDA for approval. The highest success rate is at 26% for hematological related drugs. Excluding cancer drugs, which make up 31% of the drug programs that are in development, the overall success rate increases to 11.9%. Phase 2 trials are where most drugs fail. Only 31% of drugs that enter phase 2 go on to phase 3, either due to failure of the study of the efficacy or toxicity or lack of funding. 85% of drugs that are submitted to the FDA receive FDA approval. Drugs for rare diseases are far more likely to succeed, with 25% of rare disease drugs that enter clinical trials get FDA approval. 58% of drugs that begin phase 3 trials are later submitted to the FDA for approval. So let's talk about each phase and the importance of it. Phase 1. For cancer, it is to determine the maximum tolerated dose. For other therapeutics category, it's to determine a recommended phase 2 dose. Characterize the side effects and DLT. Understand the pharmacokinetics and pharmacodynamics. And one question that's still out there is, what is the starting dose for the phase 1? For cancer or cytotoxics, it is 1/10 the LD50. For other therapeutics classes, it is different. For most therapeutic classes, cardiovascular, infectious disease, etc., phase 1 studies are done in normal volunteers. Okay? That is not the case for oncology. Phase 1 trials are done routinely in patients with cancer and routinely for those that have failed all other standards of care for their type of tumor. Phase 2 trials determine the efficacy in different tumor types or individual populations. Refine the pharmacokinetic data. Phase 2s are typically single arm, typically single institution, and you're trying to maximize the chance of detecting a clinical response or biological activity. We typically use a Simon two-step approach, looking for 1 in 12 to 15 patients. If we see that have activity, if we see that activity in the first 12 to 15 patients, expand it to 30 to 40 patients. If we don't see activity in the first 12 patients, there's only a 10% chance of rejecting a drug that has a true response rate of 20%. Okay? So the chance of rejecting something that is active is pretty low. Now, there's no standard formula for making phase 3 go, no-go decisions. You have to put together all the data and realize the next step is going to be very expensive to do and time-consuming. So, phase 3, hundreds of patients, randomized, multi-institutional, and today typically multi-national. Response intensive. Control group is usually receiving standard of care plus placebo compared to standard of care plus the investigational drug versus the investigational drug. Okay. Broad selection of patients to represent the community. Again, for oncology, the endpoints are slightly different. The endpoints for oncology, time to progression or progression-free survival. But the real standard here is overall survival. Can we prolong survival? Relief of symptoms and a delay in event. Orphan drug status. Drugs intended to treat diseases affecting fewer than 200,000 people, or if it's more than 200,000 people because of concerns that the cells won't be able to recoup the development cost, okay? But it's usually a design for a small population, gets orphan drug status from the FDA, which allows longer period of market exclusivity. Types of approval from the FDA. There's regular approval and accelerated approval. Regular approval, direct evidence of clinical benefit, or improvement in an established surrogate marker. Accelerated approval, surrogate endpoint likely to predict clinical benefit, and with that, you have to follow up with a phase 4 trial. Accelerated approval, intended to make drugs available quickly for individuals or diseases that are very life-threatening. Approval based on preliminary evidence such as surrogate marker changes prior to formal demonstration of patient benefit. As I mentioned earlier, phase 4 trials. Post-marketing, so you could have received FDA approval, but pharma uses it also to expand market indications, and it's required for drugs that receive accelerated approval. The median duration of conducting a phase 4 trial is 53 months. Let's discuss translating clinical trial results into the clinic. Tannick published a paper, which I think is excellent, that compared the overall survival of docetaxel plus prednisone for their routine patients that they were seeing in their hospital compared to those individuals that were enrolled on a clinical trial. 314 patients at their institution received docetaxel prednisone for the treatment of prostate cancer. 43 patients at their institution were enrolled on a clinical trial. Within that population, the overall results were 13.6 months for the routine patients, the standard ones that came to the clinic, versus 20.4 months for those individuals that ultimately enrolled on the clinical trial. So you can see the difference that the published clinical trial results probably don't reflect what you're going to actually achieve within the clinic routinely. Tax 327, which is the paper that appeared, the clinical trial that appeared in New England Journal, had an overall survival of 19.2 months. If they limited the routine patients to eligible criteria, it did increase it to 16.7 months, but still never achieved what appeared in the publication. Clinical approval success rate for all anti-cancer agents is 13.4 months. Small molecules, 14.3, large molecules, 11.5. Agents developed for hematological malignancies had a higher clinical approval success rate. Success of second or third indication depended upon the first indication. If the drug did not receive approval for the first indication, the chance of receiving second or third indication is very low, 2.5% and 1.8% for second and third respectively. 4 main reasons a drug fails: lack of efficacy, side effects, the pharmacogenetics, and the pharmacokinetics. Poor bioavailability, poor half-life, protein binding concerns, drug interaction concerns. The historical 3 principles of pharmacology were defined in the 16th century. Each disease has a specific cause for which there is a specific remedy. Each natural remedy has an identifiable component. The size of the dose determines the degree of response. Paracelsus was a Swiss physician and is classified as the father of toxicology. He created the dose makes the poison, the concept that distinguishes pharmacology from toxicology, and is really the basis of dose-response relationships. He also said all substances are poisons, depending upon the right dose. So the difference between pharmacology and toxicology. Pharmacology is the interaction of chemicals and macromolecules with the biological system to yield a therapeutic or beneficial effect. Toxicology is the field of science that helps us understand the harmful effects of chemicals. Substances, situations can have on people, animals, or the environment. In medicine, a side effect, whether therapeutic or adverse, is secondary to the one intended. Okay? So let's look at Viagra, for example. Viagra was synthesized by Pfizer and was thought to be effective in the treatment of hypertension and angina. A phase 1 clinical trial was conducted with the drug and was determined to have no effect, no cardiovascular effects. However, they noted marked penile erection. Ultimately, the drug received FDA approval for that indication. So let's talk about adverse events. The adverse events criteria were really set by the National Cancer Institute that went through and graded all potential side effects, both laboratory and clinical symptoms. Grade 1, mild adverse event. Grade 2, moderate. Grade 3, severe adverse event. Grade 4, life-threatening. Grade 5, death. So let's look at a few examples here. Liver function test, ALT. You can see that in the printing here, it may be a little small. Grade 1, 2.5, the upper limit of normal. Grade 2, 2.5 to 5 times the upper limit of normal. Grade 3, 5 to 20 times the upper limit of normal. Grade 4, greater than 20 times the upper limit of normal. Grade 5 is associated with death. So let's look at symptomatic clinical use of these grading criteria. Cystitis, going from grade 1, asymptomatic, all the way up to significant bleeding in the urine. Okay? Grade 5, obviously death. Every drug has a therapeutic window. Most therapeutic categories, this is very large. For cytotoxic or chemotherapies, we tend to push the upper limit of normal. So there is a line, a point, where we go above it, we increase the risk of toxicity. There's also a line or a point, plasma concentration curves are shown here, where if we don't get above that, we have minimal chance of having efficacy. And that is called the therapeutic window, that concentration range between those two. Okay? Again, for oncology, we tend to push the upper because most of the drugs have not been that effective over the years. So let's look at the drug exposure effect relationship. A dose or a drug comes into the body, input it. It has to be absorbed if it's oral. Intravenous ejection is the other potential route, as well as all the other ones we could think of. It's distributed throughout the body. It starts being metabolized and eliminated at that point. This is what we characterize as pharmacokinetics. The other lectures throughout this course will delve heavily into each of those processes. Then the drug has an effect. It can either be a beneficial effect, biological effect, or toxicity. That is the pharmacodynamic effect. It's important to realize genetics play a role in all of these processes, whether that is at the transporter level or the metabolism level or at the site of the tissue or the receptor as to whether the drug will work or not. Let's turn to pharmacodynamics. Most drugs work via receptor bonding. Okay? Drug receptor or drug target is a cellular macromolecule complex which the drug interacts with to elicit a response. Drugs commonly alter the rate or magnitude of the intrinsic cellular response rather than create a new response. Let's turn to occupancy theory. The response of a tissue to a drug, exogenous, xenobiotic, or a ligand, endogenous or exogenous, is a function of the number of receptors that are occupied. Site on receptor where a drug bonds is the bonding site. The concentration of the drug is an important factor for the extent of the receptor bonding, and the affinity for that receptor is important. Typically need to reach a threshold of receptors to get the effect you're after. Agonist effect, positive effect on the receptor. Drugs that bind to physiological receptors and mimic the regulatory effect of the endogenous ligand. Primary agonist is a drug that bonds to the same recognized site of the receptor as the endogenous ligand. Allosteric agonist is a drug that bonds to a different region of the receptor to induce its effect. Allosteric agonists still mimic the effect of the primary agonist. It's just bonding at a different part of the receptor, the protein. Antagonists, drugs that block or reduce the action of the agonist. And there you can have competitive, non-competitive, or functional. Competitive competes directly with the agonist for the same site and overlaps the receptor. Non-competitive interacts with a different part of the receptor. And functional, indirect inhibitor, the cellular or physiological effects of the agonist. So let's look at one example here. The androgen receptor. The androgen receptor, testosterone, comes into the cell. By 5-alpha reductase, it's converted to DHT. What is a 5-alpha reductase inhibitor? Enzyme inhibitor, finasteride. So DHT then bonds to the androgen receptor, which is stabilized by heat shock proteins, and then translocates into the nucleus to bond to the androgen response element. Now, we had 3 known androgen receptor antagonists: flutamide, bicalutamide, nalutamide. All of them were not very specific, not a high affinity for the receptor. More recently, a drug, enzalutamide, has received approval. This is looking at 3 Kaplan-Meier curves from the post-chemotherapy development program for enzalutamide. The trial was called AFFIRM, but just look at the A panel there. Overall survival was enzalutamide versus placebo. Post-chemotherapy, post-docetaxel, the overall survival benefit was statistically significant towards enzalutamide. And again, all this drug does is bind to the androgen receptor to prevent it from translocating into the nucleus. So it's a receptor antagonist. Now, we can have partial agonists as well. They cannot produce the maximum response of which the tissue is capable, even when it bonds to the same number of receptors. Drug properties. Specific binding of drugs to receptors depend on the physical and chemical properties of the drug. Important factors include the pKa, conformation, and stereochemistry. Warfarin, for example, is a racemic mixture of S and R enantiomers. S is 4 times more potent than R. Other examples include antiarrhythmic drugs and antihypertensive agents. Another example is thalidomide. Thalidomide, everyone remembers the tragic story of some 10,000 infants being born with a birth defect in the late 1950s, early 1960s, primarily in Europe, Canada, and Australia, from the mother taking as little as 1 single dose of thalidomide. The highest risk for the teratogenicity complications were when the drug was taken between week 3 and 8. And it turns out the R enantiomer is probably what results in the pharmacological effects of thalidomide. However, the S is probably what results in the teratogenicity. Receptor properties contain hydrophobic, often inside protein or the lipid biolayer, and hydrophilic, protein exterior segment of the protein. Binding site possess unique chemical properties based upon unique folding of the protein. Drug-receptor bonds. There were really 4 types: van der Waal, hydrogen, ionic, and covalent. So let's look at a few examples there. Imatinib, or Gleevec, interacts with the BCR-ABL kinase. Okay? It fills in the space of this kinase. There you can see the van der Waal interactions that occur in the panel B. And then in panel C, you realize that it prevents phosphorylation happening, which results in the inactivation. Gleevec was a very special anti-cancer drug that was very, very active and was noted in early phase 1 and phase 2 trials. One of the first publications appeared in New England Journal for the treatment of CML. And these are the Kaplan-Meier curves, and you can see the imatinib versus the standard of care at that time. It quickly became the standard of care and has really prolonged survival for this disease. Let's turn to sorafenib. Sorafenib is a TKI that has effects on a lot of different kinases. Okay? Now, the important thing here is to look at the IC50. And you can see the range of activity, the potency for each one of these kinases, okay? RAF1, for example, 6 nanomole. We can go over to VEGF2 at 90. FGF, for example, 580 nanomole. So in certain kinases, sorafenib is much more potent. And this is what we have seen with all the different tyrosine kinases. They have different activity against each of these growth factors or kinases. With that, there's been different clinical activity as well. One, sorafenib, hepatocellular, as well as renal cell cancer. It has received FDA approval. Drug-receptor interactions. It's important to understand affinity, intrinsic activity, selectivity, and numbers. How many receptors are available for binding? Affinity, how well the drug bonds. Intrinsic activity, produce a measurable effect. Selective, intended target. Is it hitting the intended target? So let's look at the adrenergic receptor selectivity. Agonist versus antagonist. You can see different drugs listed there. Dopamine, for example, phenylephrine on the agonist side. On the antagonist side, you can see a list of drugs listed there. And depending upon which one they're trying to hit, the alpha or the beta receptor, alpha 1, alpha 2, beta 1, beta 2, beta 3. Induced fit binding. Binding of a drug to the receptor results in a conformation change in the receptor that enhances the affinity of the drug for the receptor. This goes beyond the static lock and key idea of a drug hitting a receptor. Change in the shape induced by the drug is often identical to what happens with an endogenous ligand. Insulin, for example, is a good example of this. Next, let's turn to some of the chemotherapies and how they actually work. Platinum is a great example in that it forms these adducts in the DNA, okay? And this prevents replication. This platinum bonding adduct prevents the DNA from breaking and dividing. Next, let's turn to examples of drugs that inhibit transporters, okay? And a lot of drugs inhibit this. And the need to understand the Ki. The Ki is the inhibition dissociation constant. So this is looking at data from my laboratory of a drug that was identified through a high throughput screen at the University of Kansas, uracilic acid, as an inhibitor of OATP1B3. We used 3 substrates, testosterone, dihydrotestosterone, and androstenedione, and characterized the Ki for each of these, okay? So was uracilic acid able to block the transport in of these substrates? Rational drug design. The application of structure-activity relationships to develop new drugs with improved pharmacological effects, okay? It's been around for years. I mean, one of the first was the barbiturates, okay? Patented by Bayer, used for sleeping aid in the early 1900s. But it's the idea of changing the structure in order to, or designing something to actually hit the intended target. So I'm going to go back to thalidomide because I think it's a good example, that a drug that has a lot of different activity. For example, it's known to affect T cells. Ultimately got FDA approval for the treatment of multiple myeloma. It was initially developed for sleep aid, okay? But it's affecting cytokine productions. It's affecting COX-2, TNF, and an inflammatory cascade, and it also has effect on cereblon. With all of these different activities, we asked, could we identify drugs that were more specific for one of the molecular effects? So we have synthesized a large number, over 300 analogs. And this is just one example of how we go about trying to identify one with enhanced anti-angiogenic properties, using a zebrafish model that measures the length of growth of the blood vessels, putting the drugs, the different analogs in the water. Were we able to block that expansion or that growth of the endothelial cells? The highest anti-angiogenic had the shortest vascular effect and the fewest vascular numbers. Those with less effect were farther along on these curves. We did the same thing, trying to optimize the anti-inflammatory effects using GFP-labeled neutrophils and looking at the recruitment to a slice in the zebrafish tail. And then we labeled or plotted out all the different analogs as to how they were able to affect the recruitment of those neutrophils towards that injury. Ultimately, we came up with this type of chart, trying to, of the 315 analogs we developed, on one side, what were the most effective for inhibiting angiogenesis. On the right-hand side, what were the most effective at having anti-inflammatory activities? We narrowed that down to 16 analogs that were anti-angiogenic, 10 that were anti-inflammatory, and 3 that had effects across both ways. Then we took those 29 compounds and have taken them into models to try to develop the best one and identify a lead compound. But most importantly, with all of those 315, we've been able to develop a structure-activity relationship so that we can then determine what side chains are necessary for what activity. And this is the approach that is really powerful as you then can predict and develop new agents. The FDA ultimately approved 2 analogs of thalidomide, both of them for the treatment of multiple myeloma, lenalidomide and pomalidomide. I hope you've enjoyed today's lecture. Clearly, we've not discussed all the mechanisms of action of all the drugs. There are more out there that work differently. I tried to give you an overview of pharmacology and how it pertains in the drug development process. Nonetheless, if you have questions, please contact the course coordinator. Thank you for listening.